So what we are presenting today is the updated analysis on the ZUMA-1 trial, which is a trial evaluating CD19 CAR T-cell therapy with axi-cel or UCell in patients with refractory aggressive B-cell non-Hodgkin lymphoma. So we have a median follow-up of 15.4 months in 108 patients at this time, and we observed that the best overall response rate in these patients with a single infusion of this axi-cel therapy is 82%, and the best complete remission rate is 58%. And at 15.4 months, we find that 42% of those responses are ongoing, with 40% being in complete remission. So this therapy does not require any second infusion, so all of these responses have been observed with a single infusion of this treatment. So that's what is quite remarkable about this. Longer follow-up is ongoing, but we do have longer follow-up on earlier single institution phase 1 trials where this product was initially developed at the National Cancer Institute in Bethesda, Maryland. And they have long-term follow-up with the longest patient still in remission, more than 7 years out, and many patients in remission more than 4 years out. So suggesting that these responses can be durable, and maybe a significant proportion of these patients may have even a cure from this refractory B-cell lymphoma. Yeah, so we do have a number of different ongoing trials and planned trials. So at this time we are trying to see if we can further improve the efficacy of this product by doing a combination trial with axi-cel in combination with anti-PD-L1 antibody. So that data is being presented at this ASH meeting as well. In addition, we are also evaluating axi-cel in other B-cell lymphomas such as mantle cell lymphoma and indolent B-cell non-Hodgkin lymphoma, as well as CLL and acute lymphoblastic leukemia for both pediatric and adult population. In addition, in the next year, we plan to initiate a randomized phase 3 trial to directly compare the efficacy of axi-cel with autologous stem cell transplantation in second-line setting in patients with refractory diffuse large B-cell lymphoma. So I think it's been quite difficult to identify a biomarker that correlates with the response. What we've done on this study is to go back and analyze patients who have progressed or relapsed after axi-cel infusion. And in a very preliminary data set of about 21 patients where we analyzed, we observed that a third of these patients actually have loss of CD19 at the time of relapse. And 2/3 of the patients have upregulation of PD-L1, suggesting that a combination of axi-cel with PD-L1 might make it more effective. But in the patients who have loss of CD19, we may have to use bispecific CARs or dual CARs to target more than one molecule besides CD19.